Hi, I'm Sean Mooney, professor at the University of Washington and Chief Research Information Officer of UW-Medicine. Over the past 2 years, we have been creating an initiative in medical data science, and as part of that, we have created this seminar. Speakers will include world-leading scientists who will tell us about how data can impact human health and their healthcare. Please use the link below to register for our mailing list to hear about future events in this series. We hope you enjoy them.
So now I want to introduce Dr. Ali Shojaie. He is the professor of biostatistics and adjunct professor of statistics here at UW. He is the associate chair for strategic research affairs at the Department of Biostatistics here at UW. He's the founding director of the Summer Institute for Social Sciences. He is the affiliate faculty member of 4 different institutes at UW: the Center for Statistical Social Sciences, the Biostats and Biomathematics portion at Fred Hutch, the eScience Institute, and as well as the Algorithmic Foundations of Data Science Institute. His research focuses on the analysis of large complex systems, particularly biological and social systems. And today he's going to be giving us a talk about the explainability of machine learning. So Dr. Shojaie, I'll hand it over to you and you can share your screen.
Thanks, Noah. I'm glad to be here. I only make one quick correction that I'm the founding director of the Summer Institute for Statistics of Big Data, not social science. That's a different one, but I'll get to that at the end. So I'll share my screen. Sorry. So I want to talk about sort of some of the work that we are doing and we've been doing in my lab, but more broadly, maybe a little bit about, as Noah mentioned, explainability in machine learning and from a statistician's perspective. So I'm a statistician by training. I came to statistics and eventually biostatistics, starting as an engineer, but found my way to initially statistics, and then along with my PhD in statistics, I also did a master's degree in human genetics, and that's what when I really got interested in applications in biology, in particular in studying biological systems. So my talk will focus on explainability in biological systems, especially when we want to use machine learning for understanding how biological systems work. So I don't need to say much about all types of data that are being collected in this seminar and to this audience, but basically we're interested in a variety of phenotypes, and we know that both genetics and genomics, but also lifestyle and environment affect our various phenotypes. And to understand human health, we need to understand both of these aspects and a variety of variables that might influence our health. And we're collecting more and more data, increasingly more data on various aspects of our lives, from genetics and genomics to down the biological pipeline to proteomics and metabolomics and microbiome data. On the other hand, we're also collecting imaging data, EHR data, data on our activities through sensors. We're collecting data on environmental exposures, etc. So there's a wealth of data out there, and lots of opportunities for using machine learning approaches for understanding how these various biomarkers are associated with our health, and how we could develop new treatments potentially. What I am interested in, broadly speaking, and some of the work that we've been doing in the lab is this intersection of machine learning and AI with biology of medicine, and there's a lot of applications where we use machine learning tools or AI tools to make predictions. So especially with the rapid increase in computing power, now we're using more and more deep neural networks to make better and better predictions. So we pass a bunch of data. This could be EHR, this could be sort of imaging data, various types of data that we are passing to these machine learning approaches, including sort of deep neural networks to get better predictions. So we get improved prediction potentially compared to classical approaches. But then sometimes it's difficult to obtain an insight into what those predictions mean and where those predictions are coming from. This has been an area that has been quite active research area over the past 5-7 years, and people have been coming up with new ideas for how to interpret these predictions that machine learning methods are giving us. So I took this picture from a recent paper in Nature Communications that basically they propose an approach that takes that deep neural network and basically then goes back and tries to come up with an explanation for different variables. So instead of knowing which variables are included, it gives weights to these different variables, saying that maybe the past value of creatinine is most important in forming this prediction. And as I mentioned, this is an area that has been quite hot over the past 5 to 7 years, with a lot of interesting work done in understanding what machine learning models do and how we could obtain insight into the prediction that these models give us. And this is critical because especially in clinical applications, even though there are a lot of applications where prediction is important, in many other applications that prediction might not be enough. You might need to know something about the variables that are involved in making that prediction, or more importantly, in some cases, we might need to, when we want to develop treatments, we want to go beyond just prediction. We want to answer causal questions. So this comes up naturally in biomarker discovery and treatment development, and many other applications. And as I mentioned, this is a growing area of research, including some interesting research by colleagues here at UW. And we've also been, with my colleagues in the Department of Biostatistics, and also some of my colleagues in Biostats independently have done work on this problem. Naturally, the work that has been done in Biostat and Stat departments has been more focused on statistical measures of importance and statistical approaches to variable importance, whereas some of the work that has been done in CS has focused on alternative approaches for variable importance. I see that there's a question. I don't know if we are taking questions now. I'm happy to take questions now. But Noah, you're the boss here. Tell us how we should proceed with questions.
I think we'll try to accumulate some questions and we'll do them at the end, if you don't mind.
I'm totally fine.
Great.
Yeah. Okay. So maybe please, if you could type the question in the chat, that's what the message was that Sean had sent, and then we'll get to these questions at the end. Okay, so there's a growing literature, as I mentioned, on trying to explain these ML and AI algorithms, and that's what I'm interested in. The more specifically, what I'm interested in are settings where, unlike classical applications of these ML and AI methods, which require lots of samples, might not have lots of samples. So classically, sort of AI and many ML methods work in this setting that we have few variables. So these are the columns in my data matrix, and lots of observations or samples. Whereas there are settings where we have lots of variables and maybe not as many observations, and so this small sample setting is something that could become challenging for some of the AI methods, and naturally for explanation methods, this might also become somewhat difficult. So that's an area that I'm interested in, and I've been working, and I'll talk about this particular area a little bit. So I want to talk about explainability for biomedical data, but a lot of my big data are big data that were the number of variables might be larger, not necessarily the number of observations. So, but nonetheless, we want something that works irrespective of the number of observations, that works both with small data and large data. And a lot of the data that I'm interested in, and I'll talk about a few examples, are data that have complex structures, that either we're collecting data that is over time or over space, or data that is being collected that comes with additional structure that we need to take into account. So we want to incorporate that biological structure and additional structure when analyzing this data. And again, I'll talk a little bit about a couple of different examples. And the goal overall is to provide mechanistic insight into how the variables are related to the outcome and with each other. And that is what I'll focus on today. I should mention that I'm most likely, and excluding many relevant references here, this is just a small list of many of the references that have been popping up in this area. So there's a growing literature on this. In case you're interested, I'm happy to provide additional references. And in terms of application areas, I'm going to focus primarily on neuroscience applications today. I'll talk briefly about some genomics ideas as well, but the ideas are broadly the same. It's the same types of tools and methods that we're using with some small tweaks. And I'll talk about methods that we've used that were originally developed for genetics and genomics that we've taken them to analyze neuroscience data and vice versa. So you see that hopefully the core ideas are not that different. It's just that the general idea of incorporating the external structure and information, and also trying to come up with explanation and mechanistic insight as we are dealing with these problems. Okay, so a big chunk of what I've been working on over the past few years has focused on understanding the brain connectome and using the information that is in the connectome to understand how the brain functions, what goes wrong when we have neurodegenerative disorders, and hopefully use that information to design new treatments. I'm going to go through a couple of examples. I won't go deep in any of these because it might get technical, but hopefully that'll give you a flavor of the type of questions that could be answered using these machine learning tools when we think about explanation and we think about mechanistic insight. Here's a first example when in collaboration with Yaz Dunlap, which is at bioengineering here at UW, and colleagues at Applied Math and Stat Department, and two really talented grad students. So we've been working on figuring out how to use the connectome, the brain connectome, in order to inform neuromodulation. So the idea is that, and this is what the Yaz Dunlap has significant expertise in, they have developed these ECOG arrays that allows them to not only observe the activities of many brain regions, but simultaneously, but also be able to use laser and in conjunction with optogenetics to intervene at let's say two locations on the brain. And this is done in monkeys. And by doing this, they could basically send the laser pulse and see how the monkey's behaviors change. And obviously, this could have various implications. This technology has been quite interesting for many applications, and it's being used for a lot of neurodegenerative disorders. But the goal here is that we need to understand this effect of this neuromodulation in order to be able to use this technology more effectively for therapy. Right now, even though sort of neuromodulation is used, in particular, it's used in schizophrenia and depression, it's used without being able to pinpoint the effect of neuromodulation and the laser treatment. And the goal of this study was to see whether we could get more insight into the effect of these, the effect of neuromodulation. So here's a quick schematic of the experimental setup that, as I mentioned, Yazdan Lab has developed. They have ECOG arrays that measures about 100 electrodes that sort of are measuring activities of at the brain surface, and these measurements are obtained during laser stimulation, and they predict changes. They basically induce changes in connectivity, and the goal is to be able to predict the changes in connectivity using features of the experiment, usually features of the protocol features, that basically the location of the lasers, their distances from each other, etc. But also what we're trying to do in this particular project is to use the connectome features before stimulation to predict how the connectome is going to change after stimulation. And the idea is that the connectome is going to be predictive of of various functions. If we could predict how the connectome changes in response to stimulation, might be able to predict how it would also respond to affect the function. So that's, however, that's another layer that we're currently working on. This piece, I'm only going to talk about predicting the changes in connectome as the result of this laser stimulation. And in general, the way that this is set up is that there are these intervals when we record, and then we test at baseline, and then we have laser treatment, and you see that sort of the different lasers with some frequency are applied, and then we record and test again, and sort of we repeat this experiment multiple times, and that's the data that goes into figuring out how the connectome changes after the stimulation. Now, we measure the connectome using basically functional connectivity. We look at the functional connectivity that is quantified by coherence. And the coherence is basically calculated at multiple standard frequencies that are of interest in the field. These are different frequency bands, from theta band all the way to high gamma band that potentially you get at different levels of spatial resolution. And these are calculated over the entire network, not just for the association between the laser locations and the other measurement locations, but we obtain a whole network. And then we look at how the coherence has changed before laser and after laser. And the goal is to predict this change in laser coherence. So what the Yazdan Lab and others in the field had done previously was that they were using these pretty simple linear models to predict the changes in connectivity. And classically in this field, people use features of the design, features of the experimental protocol to determine, to predict how connectivity could change. What we've been doing in collaboration with them has been to use basically more flexible machine learning approaches that allow us to model better model the changes in connectivity and their associations with various features, but also in a way that is explainable. So in particular, what we've been using for this project is a class of hierarchical additive models, which instead of having linear functions in each feature, have these flexible nonlinear functions in each feature. And that together, they allow us to model very complex surfaces. However, each one of these functions is developed based on a class of hierarchically more complex functions of the feature. So for example, this one is constructed by considering linear and quadratic and cubic polynomials. So we have these increasing levels of polynomials that make up each one of these features. And the polynomial degree is automatically decided by the model. And we end up with a model that is both flexible to capture many different types of relationships between the coherence change and these features, but also in a way that allows interpretation of the association of each one of these features with the changing coherence, which is important for developing therapies and for understanding how the features impact the changing coherence. And this is based on earlier work that we had done with one of my former students. And the results were quite interesting, both in terms of how the network features, the connectome features are informative for predicting changes in connectome, but also how using a more flexible yet explainable machine learning approach can allow us to gain insight into what the effect of these features are, or what the association of these features are. So here's a plot that shows basically if we use protocol features alone. So this is a prediction model using a flexible framework and similar to the one that I explained. Using that, if we only use protocol features, we don't do a good job predicting the change in functional connectivity. So that's the functional connectivity change. And that's shown in these very low R-squared values when we look at only protocol features. On the other hand, if we use network features in addition to protocol features, we now get much better predictions. And that's what's shown in this plot. Should probably use my spotlight here. So that's shown in this plot, and we see that sort of we get much better predictions across different frequency bands. And that's also represented in significant improvement in R-squared, and this is our test R-squared. So this is not R-squared in the training data, but R-squared in independent test data. And we see that we gain quite a bit in explaining the changes in connectivity across different frequency bands. And that's also shown in these pictures, that sort of this is the true change in connectivity in the test data. If we only use protocol features, we don't do a good job at all predicting what that change in connectivity is. We're basically predicting everything to be almost the same across the network. Whereas if we use the network features and the protocol features together, we do much better, and we get much closer to the true change in connectivity. And this is quite important because this technology, as I mentioned, has been used for quite some time now in combination of basically neuromodulation in different strategies, not exactly with optogenetics and not at the scale that the Yas Dunlap is using it. But optogenetics, but neuromodulation has been used before, and people were very excited about using this for treating various neurodegenerative diseases. And there were a couple of clinical trials that started, but they didn't get really good results, partly because they were primarily using these protocol features. And because of that, it was difficult to tailor these treatments to individuals, and the results of these clinical trials were not very encouraging. Whereas what we are seeing here, again, this is a proof of concept. This is very early work that we've started. But what we are seeing here is that by incorporating these network features and more flexible models, we are able to gain quite a bit of improvement and get much closer to the true change of connectivity, which again, change in connectivity is the first step towards trying to predict the function, and that's down the road and what we're currently working on. But at least these are much more encouraging results than what the field was getting previously. And as I mentioned, these are models that are also very explainable. Unlike a deep neural network that we could have used here, we actually gain insight into across different bands into how each one of these features is associated with the change in connectivity. So for example, distance between the laser sites, how that distance is associated with changing connectivity. And you see that function is learned flexibly from data is quite similar across bands. So we get confidence intervals for these curves, and basically we get to see how change in distance would affect the change in connectivity. And these are, again, sort of across different frequency bands. These are observed improvements compared to a linear model that is the baseline that the field uses. And so this comes again from looking at both these network features and these protocol features in a flexible model that is by design explainable, because by design it allows us to determine which order of polynomial is relevant. And because of that, we could both get insight into the relationship between each one of these covariates with the outcome, or with basically changing connectivity, but also the degree of polynomial is chosen automatically from data. So that allows a flexible fit that provides some explanation. I won't go into details of how we chose these various network features. This is somewhat standard in network analyzing network data. So I will not go into these, but I'm happy to discuss them later if there are any questions. So this was one example of the type of analysis that we could do. And here we were interested in changes in functional connectivity and developing flexible machine learning methods for understanding this change in connectivity. Now, the connectome itself here, we learned it using coherence, which is a notion of correlation. This correlation, however, and this measure of functional connectivity is somewhat limited in the sense that it captures indirect associations between two locations on this ECOG array. That means that if two locations are associated, both of them or correlated with the third one, they would also be deemed correlated. And this could be misleading. So this is a joke that I always throw in my slides whenever I have talks that computer scientists are also in the audience. And this is historically looking at the association between the number of CS PhD awardees and per capita cheese consumption. And this is not fake data. This is data that if you just look at correlations, something naturally pops up. But this is because, as I mentioned, correlations could be misleading. That if you have two variables that are correlated with a third one, let's say in this case, correlation between both x2 and x3 with x1 is 0.8, then x2 and x3 by definition will have a correlation of 0.64, which is not trivial correlation. So you will end up deciding that these are also correlated with each other. And that's the limitation of correlation. So what we've been working on quite a bit has been alternative approaches for understanding connectivity that are less misleading and better are better able to capture the relationship between different features in our data. So for example, we could estimate these networks using partial correlations instead of correlations, which allows us to get a better sense of the association between variables with each other. And so I'm going to talk a little bit about how we could use these alternative measures of association for understanding the connectome. And in fact, what I'm going to talk about is also a class of approaches that allows us to take into account the temporal patterns of activity, not just partial correlation, which is correlation adjusting for the other variables, but also taking into account temporal patterns of activity across time. And this is a notion that comes originally from econometrics. And I'll talk a little bit about this idea of using what is known as Granger causality in order to get better estimates of connectivity. So this notion, as I mentioned, comes from old work in economics literature, and Clyde Granger back in 60s came up with this idea that he called a time series X causal for another time series Y if past values of X could predict future values of Y. Now, I'm calling it Granger causal because as you can see, and I've put that in italic here, it's all based on an idea of prediction. In econ literature, there's still debate on whether this could get you into causal insight, and I'll talk a little bit about that a little bit later in the talk. But in general, this is purely based on prediction, so we can't expect it to be providing any perspectives into causal relationship between variables. But this is an approach that has been quite popular and has been used a lot across different fields. It came from economics, but has been used in neuroscience quite a bit, and in other areas as well. And the idea is that, for example, here's two time series, one showing COVID-19 cases around the beginning of 2020, and then showing the response of S&P 500. And we see that as COVID cases started going up, initially, the stocks took a big hit and went down, whereas then as people perhaps understood how this will impact and what the magnitudes of it are, the stocks sort of went back up, and we've seen the behavior of these stocks. So the whole idea of Granger causality is to use these changes of variables over time to see if one could predict another. In this case, whether changes in COVID-19 cases could predict stock prices. But more generally, what I could explain is that suppose that you have two variables, two time series X and Y, and you're interested in predicting the future value of time series Y. So this is what you want to predict. You could do two things. You could ignore X completely, and then that way you might get this prediction for future value of Y. Or you could use the value of X in addition to the past values of Y in order to get a different prediction. And the whole idea of Granger causality is that if that prediction is better than the one that you would have gotten only Y, past values of Y alone, then you would call X to be Granger causal for Y. And this basically is a notion that then can be extended actually to multiple variables, not just two variables. So for example, we could look at a notion that we've called network Granger causality. And this is an extension of this idea of Granger causality to many variables. So this could be genes or regions of interest in brain, etc. And if you have, for example, in this case, you might have three genes that the first one positively affects or is associated with gene 3. The second one negatively affects or is associated with gene 3. And then we could look at the time series of genes 1, 2, 3 over time, or brain regions over time. And again, these ideas could work in different domains. And then maybe zoom in a particular area, and then we look at that region. Maybe we look at the expressions of genes 1, 2, and 3. We see that as this is the expression of gene 1 and 2. And then if we look at gene 3, we see that as gene 1 goes up, because it positively impacts gene 3 goes up. But as gene 2 goes up, because it's an inhibitor, then gene 3 also goes down. So those are the types of behaviors that we could use in order to predict which genes are associated with others. And basically would be able to construct this network from observations on these nodes. And this is something that people have used a lot, as I mentioned. So there's a rich literature on using these ideas for learning brain connectome, for example. But the vast majority of approaches that are out there either use these simple linear models or use pairwise models that ignore the other variables. So they don't do this Granger causality learning at the level of the whole network. They only look at 2 variables at a time. And that's limiting in the sense that it could, as I mentioned, it could be misleading. It could miss associations with other variables. So what we've recently worked on has been a new approach that allows us to do this, infer these connectomes using deep neural networks. So we have brain regions that we collect signal at these brain regions, and we pass these brain regions to a deep neural network, and then we try to identify Granger causality. Now, this is my attempt at having slides without any formulas failed in this at this point. That these are the 2 slides that have like a little bit of formula in them. That here, a neural network, what it would do is that it would basically model the activity of genes at time t based on the activities of past genes, all of them together. So you would look at all the previous time points, and then you fit a model, sort of a black box model, really, and then you try to predict what x at time t is. However, that will be difficult for explanation, in particular for inferring this connectivity network, if we were to use this notion of prediction for Granger causality. That would not work. So instead, what we have worked on has been to develop a regularized class of deep neural networks specifically for Granger causality. And I'll try to briefly mention this idea, how this works. There are 2 pieces into the working of these regularized deep neural networks. The first part is that these are component-wise. So what do I mean by that? Instead of modeling the whole vector, we look at each one of these variables at time t, and then we try to model them based on the previous observations. And they're also regularized in the sense that there's a sparse input layer in these networks. So what this does is that, and these are 2 examples. One is with a multiple layer perceptron, and the other one is with a schematic of a recurrent neural network in LSTM. And both of these are component-wise, and as you see, they model the variable I at time t as a function of all the previous variables in both cases. In the case of MLPs, these component-wise MLPs, we add sparsity-inducing penalties at the input layer so that the past values of some of the variables are either included or excluded in this model. And this basically means that we have different penalties, and that's what this schematic in the middle shows, that we either have these group sparsity penalties, or that we have these hierarchical penalties that allow selection of the lag as well, of how many time series in the past should be included. Or in the case of recurrent neural networks, again, we include a sparsity at each layer, at the input layer, for each variable, in order to decide whether that variable should be included or not. The benefit of the RNNs, and in particular these LSTMs that we use in this case, is that they deal with long-range dependence. So we don't have to worry about selecting the lag of the time series that is relevant for constructing these connectome measures. So that's again, a modification of a well-known class of machine learning AI tools that now allows us to have a method that is explainable by design, because it has this sparsity feature built into it that selects which variables should be predictive of another variable. And the benefit of this sparsity is that it also works in cases that we don't have lots of samples, that unlike classic RNNs or MLPs that require large sample sizes, these methods also work with quite small sample sizes. So in fact, here's an example from looking at the DREAM challenge. This is the DREAM 3, I believe, where we try to infer gene regulatory networks based on time course measurements of the genes. This is, of course, a challenge to simulated data, but we had 100 genes and about 1,000 time points. In fact, not 1,000 time points, but this was like 46 replicates and 21 time points. And this is a very complex task, partly because there are hidden variables that were not made available, etc. But we see that across 5 different learning tasks, these sparse input neural networks do much better than all the other approaches that people have tried before for this prediction task. And these are sort of area under precision recall curves that we see that they're much better using these sparse input neural networks. And these approaches, as I mentioned, could work in other settings as well. So for example, one application that we looked at and I thought was an interesting application is this setting where we looked at human motion capture data that CMU had made available. And this is basically movement videos that you could then analyze to figure out how movements of different body parts are associated with each other. And we could get these estimates of connectivity between different body parts. That of course, one thing that I didn't really talk much about is that these connectivity estimates depend on a tuning parameter. Like any other regularization approach, we would need to take into account these tuning parameters. So that's something that, if you're interested, I'm happy to talk about maybe at the end. But it's something that impacts the performance of these methods potentially. Okay, so this was another approach that I mentioned, these neural Granger causality for estimating these connectivities. This works well in the case when we have continuous measurements, for example, from ECOG arrays or from fMRI data. But there are many other types of data that we are also collecting about activities in brain. So one type of data that I've been really excited about over the past 5-6 years is data coming from these calcium fluorescent imaging technology. I'm going to show you a quick video. Hopefully you guys see it here. Noah, do you see the video? Okay, I'm going to show it.
The video.
Yes, it looks good. Yep.
Okay, so you see that this is basically data that shows in a small window to the brain shows spiking of neurons. So each one of these is a neuron, and this is data from a mouse brain that you see that as the mouse is doing a task, different neurons are firing over time. And this is data that basically what they do is that they mount a little camera on top of the mouse's brain, and they're able to capture the signal live as the mouse performs these different tasks. So data after lots of pre-processing becomes something like this, that for each neuron, each line is one neuron. You have the spiking time of that neuron, when that neuron has spiked over that window that we saw in the video. So this is a really cool data set because now we have live monitoring of activities of individual neurons. And we could do this in response to task, for example, and I'll talk more about that. But this is as detailed as we could get. The technology is improving rapidly, so we're collecting more and more neurons, we're observing more and more neurons. And Allen Institute has put huge loads of this type of data, has made them publicly available in case folks are interested in it. So again, we could use these spike train data to learn the connectivity as well. And the ideas are the same, but the only difference is that now we have a different type of data. So now we're working with these spike trains that basically just tell us when each neuron is spiking. And so the idea is that we could use the past history of spiking of one neuron to see if it's predictive of spiking of another neuron. And this is quite consistent with what neuroscientists think happens in brain, that neurons respond to each other, and it could be either triggering one neuron, triggering spiking another neuron, or inhibiting spiking in another neuron. But nonetheless, we could use this data to predict spiking in other neurons. And so we've been working a bit on this area and have developed a couple methods for this. And again, sort of our goal has been to come up with measures of explanation in this particular case, whether we could come up with tests of hypothesis of whether one neuron could predict another neuron or not. And this is what we've done. I won't go into details of it, but we're able to assess whether there's a link between two neurons or not with pretty good control of type 1 error as the sample size increases, and also with pretty good power. So we see that as we have more and more data, we get close to the oracle. This oracle is basically knowing the truth. So as we could get as close to the oracle in predicting links between neurons. And this is interesting because now we could, for example, look at differences between neurons, neuron connectivities with and without laser, for example. So this is data that was collected in a paper back in 2018 by Boulding and Franks on again, mouse olfactory bulb region. And they had data with different lasers or without lasers. And when you look at the connectivity networks with and without laser, there are striking differences. And then you see that these differences correspond to basically 95% confidence intervals for these being completely separate from each other, which indicate that connectivities are different with and without laser. And that's obviously, from a neuroscience perspective, that's not very surprising. But now we have a way of measuring that and quantifying that, which is useful because in practice, what these experiments usually do is that they run multiple sessions. And this is a schematic of the way that these experiments are run. There's a camera that is mounted in a small region, and they run multiple sessions where they look at maybe different stimulus and in each experimental session, and then the interest is to see, for example, how the neurons respond to a particular stimulus, and the best way to do that is by looking at changes in these connectivity networks in different experimental sections, and so now that we have a way of obtaining these connectivity networks with confidence, then we could look at simultaneously learning these and sort of learning the differences between these networks, which is clinically very relevant because now, for example, we know that as we induce changes using laser, we could quantify the changes in these networks. And we see that sort of this actually works very well in terms of the true positives that we get from these estimates compared to what we would have gotten without these tools. And we are able, again, to control the FDR, the false discovery rate, with a pretty good power. So the power, as we increase these experiments, increases, whereas if we were to ignore this multi-session experimental setting, the power would go down, the power would diminish as we do more and more experiments. So this is another example of the type of thinking that goes into developing methods that are able to capture the complexities of these pretty complex multifactorial designs and able to provide good insight into changes in these brain connectivity networks. And here's an example of it that with the same data from Boulding and Franks, we've looked at no laser versus two different laser intensities. And we see that both laser intensities induce changes in the network, and the networks with laser intensities are more similar to each other than the network without laser. Again, not surprising. But now we could see how much the brain connectivity changes as the magnitude of laser changes, magnitude of laser that is induced changes as well. I mentioned that one of the reasons that I'm particularly interested in this spike train data is because we have live imaging of activities of individual neurons. And this provides a unique opportunity, in fact, for getting into what I mentioned at the beginning about this Granger causality framework, and that is whether we could actually understand causal relations between neurons, for example, whether one neuron spiking would cause another neuron to respond or not, or whether we could only predict. So can we go beyond prediction? Can we answer causal questions? And in general, there are two issues that make this task quite difficult, more generally for doing causal inference using these time series data. And this is more general. This is not just limited to looking at these spiking patterns of neurons, but two issues that make it difficult. One is the fact that our sampling time might be different than the true causal time frame. So you might have subsampling. And this is something that is known, and so we've also done some work on it, that if your true causal time frame is at, let's say, as milliseconds, and then you collect data at seconds, you're going to get data that is not going to be useful for causal inference. So that's an issue that could lead to incorrect estimates of connectivity. Another issue is having hidden processes or hidden confounders. So I'll talk about hidden confounders in a bit, but the really good thing about this spike train data is that it's so high resolution in time that basically subsampling is not an issue. We have live monitoring at the level of 30 kilohertz. So we don't have to worry about subsampling. So the issue that now we have to worry about, and if we could address it, we might be able to actually do causal inference, is the presence of these hidden processes. And in fact, that's a huge issue because we're only able to monitor a small portion of the brain, and we're going to miss a large number of neurons, regardless of how much this technology improves. So we have to somehow account for other neurons that we're not observing in order to go beyond prediction, in order to do causal inference. And that's something that we've been recently working on. And it turns out that, in fact, we could do this under some assumptions, and I won't go into details of this. And for that reason, we're calling this causal discovery because it relies on some assumptions. But under some assumptions, we could actually get to those causal links, even if a large number of neurons are not observed. So here's an example, again, with simulated data, that we see that this method that we've developed gets almost all of the true edges before it gets any false edges. With large enough sample, we are able to do a pretty good job discovering true causal relationship between these neurons before we make errors and include false positives. And this is something that I think as we collect more and more data, time course data now with imaging, with sensors, biosensors on our phones and Fitbits, etc. This is an area that I think would be quite interesting to look at and think about. It's not going to be as high resolution as calcium fluorescent imaging, but there are ways to get around this issue. And I think, especially if our goal is to understand mechanisms and how we might be able to impact, this is an area that I think will have a lot of activity in the next decade as we have more and more data. I'll briefly mention a couple ideas. Noah, how much time do I have?
A few minutes. I think we're concluding up here in the next few minutes. Is that okay? Next few minutes.
Okay. I'll just mention a couple of things, neuroscience stuff, and then I'll skip maybe the genetics, genomics stuff. Some of the other things that we've been related to this that we've been working on again, understanding changes in connectivity is for example, whether we could detect seizure from EEG signals. And this is trying to automatically identify change points in the connectome. So here's an example from an EEG, like an old now EEG signal. And this is an 18-channel EEG signal. And then if you look at these, the time series for these, you see that around sort of here, maybe something is happening that sort of the volatility for these time series is changing. And it looks like we have a different time series before and after this. And in fact, this patient had a seizure at this time. So we know that post-mortem, but basically the question is, can we detect this from data? And can we detect it in an automatic way? And so this is something that we've been working on. Right now, we have a method that could detect this not in an online fashion. So we're working on the online version of this, but we could detect when the seizure has happened, which would allow us to look at potentially connectivities before and after seizure. In this case, this patient had a seizure in the left frontal lobe, and that's exactly where you see a lot of differences in connectivity before and after. So there are these red edges that are specific to before seizure that are missing in after seizure. And that's where the patient had a seizure. This is really helpful for gaining insight into the effect of seizure and how it's impacted the connectivities. The gray edges here are the ones that are common between the two networks. But as I mentioned, what we're currently working on is an online method to see if we could use data to detect when a seizure might happen, which of course is again, from a clinical perspective, that could be very helpful because now we have devices that could intervene and potentially prevent the seizure from happening. I'll maybe mention this one last idea and then I'll skip the rest of the slides. That this is another idea, and this is coming from the world of genetics and genomics, and looking at changes in connectivity or differences in connectivity, for example, in this case in TBI, mild traumatic brain injury, and comparing that with healthy controls, and using differences in connectivity as a way of identifying biomarkers. This is based on work that has been going on in genomics literature, and there's a really nice review paper by Krogan and Eidecker that dubbed this as differential network analysis. So basically the idea is that you're looking at changes of the network in two different conditions, let's say in mild traumatic brain injury and healthy controls. And so what we've done has been to generalize this idea to the case of time series data observed from brain imaging. And this is in collaboration with Christine McDonald here at UW, that we've looked at differences in connectivity between these two conditions to see if that guides us to insight into how the brain connectivity is impacted by trauma. Okay, I'll skip over these ideas and maybe summarize. I could maybe summarize it by one slide with an artwork that I really like. This is by an artist named Nancy Rudin, and this is an airplane with all the working pieces of it. The only issue with this airplane is that, and the reason that it won't fly is that it's not assembled right. It's my way of thinking about sort of mechanisms and connections that are so important to understand in these biological systems. So as I mentioned, my background is in engineering, so I started thinking about engineering systems back in the days, and I still see biological systems as systems that in order to understand how biology works, we need to understand how things are connected with each other, how they work together to perform a function. And these connections do matter. And we need methods that provide mechanistic insight. If we want to go beyond prediction, we need methods that provide mechanistic insight into mechanisms of biological systems. I'll end with a little bit of a shameless plug because Noah mentioned it at the beginning that I lead this Summer Institute in Statistics for Big Data that we've had it for about 8 years now. And this year is going to run virtual. We've had it virtual for the past couple of years, initially due to COVID, but I think we've had really good success with a virtual institute. So there's some short courses. Each one of these is like 2.5 days on supervised learning and unsupervised learning that would be highly related to the stuff that they talked about here, but also on data wrangling and visualization in case folks are interested in. So I thought I'll put this one slide here. I'm going to stop there.
Thank you, Dr. Shojaie. I think we have a full room of people clapping, but in Zoom you can't hear that anymore. But yes, this sounds like a great course. People, please take advantage of this. We do have a couple questions, but please do give us some more questions as well in the chat. So Dr. Shojaie, I'll just start reading a couple questions to you and give you time to answer. Is that okay?
Yeah, sure.
So the first question is coming from Sean Mooney, who is talking about, well, you talked about the explainability of machine learning, the kind of mechanistic insights, but these results will still go out to providers and maybe even patients. Even with these types of results, do you think there are additional training, or what kind of training should people have if they're going to be even looking into these kinds of conclusions?
And that's a really, really good question. I think critical to any of these mechanistic insights, especially if they're going to be used by clinicians. So most, I mean, maybe I'll take a step back. Most of the stuff they talked about was when we are thinking about understanding mechanisms in order to develop new therapies. That if we want to deploy these to the point of care, that's a whole different ballgame. And I think that's to some extent what Sean is asking. That if we want to use these at the point of care, most important piece that we need to understand is how much uncertainty is there about these mechanisms that we are identifying. And I think that's really important aspect if we want to use these in clinic, which is why I mentioned sort of this piece about trying to get confidence intervals for estimates that we have in order to hopefully provide that for physicians, that sort of this is our in a sense, these are all estimates, right? This is our estimate of what the mechanism is, and these are confidence intervals, whether they're overlapping or not. So that needs to be taken into account when these tools we want them to be used by clinicians. So I think that's the most important piece. The second piece that I think Sean would appreciate here is that whenever we think about mechanisms, evaluating methods becomes much more difficult. That when we think about prediction, it's easy to define prediction tasks that you then go and put up competition for people to sort of compare their predictors to see which one does better. Whereas when we think about mechanisms, for example, when we think about sort of brain connectivity, we don't know what the truth is. We have a really hard time assessing these tools. And in some settings, that's why I mentioned sort of the Dream Challenge. The Dream Challenge provides like a benchmark for assessing whether you're doing well or not. In other cases, you have to do knockouts or use these in order to design new experiments to see if you could validate the findings of these predictions, these estimates. And that's a task that I think in general is quite difficult. So in order to be able to take these approaches and use them in clinic and at the point of care, we need to validate them first. And that validation by itself is quite difficult. So that's another piece that is very difficult if we want to take these to the next level.
I want to basically back up the comment that you just made. I mean, my group has been building machine learning algorithms to predict pathogenic genetic variants for the purposes of interpreting human genomes clinically. And as part of that, our methods will also identify mechanistic features that we think are predictive of pathogenicity. And we've had 2 R01s and a number of other grants and students that have worked on trying to prove experimentally that path, the mechanism that we're predicting, and it's really difficult to do. It's very difficult to do in a high throughput way.
Yeah, absolutely. I mean, and we're actually probably closer in genetics and genomics world, because now we have Casper nanotechnology that so we could do it like these high throughput screens, but in neuroimaging that's even harder, and in neuroscience that's even harder because we don't have technology to do high throughput. So one of the reasons that I was really excited about sort of this technology with ECOG arrays and optogenetics is that so it gets close to that. Now they're actually able to intervene, and there are labs that are getting close to that, but it's still quite challenging to validate what we find.
Great, thanks. We have another question more specifically on the connectome portion. They gave us a slide of slide 23, which was the reference for their question. But the question was, do baseline connectomes in some ways add information that is unique to an individual and hence improve prediction potentially?
Yeah, absolutely. I think that's the huge area of excitement potentially, because that baseline connectome is individual specific. And now we're saying that we shouldn't be thinking about sort of the general protocol, experimental protocol. We should be thinking about the current connectome in order to decide what to do for this particular patient. So this becomes more than just now thinking about generic treatments. It becomes much more personalized treatment that is targeted for each individual. So we need to first understand the connectivity, the current state of connectivity for that individual, in order to figure out how to alter it. And not only that, then this becomes a dynamic treatment regime, because now we have to think about how to continuously update our baseline connectivity estimate in order to then decide how to continuously change it. So I think this is I'm personally very excited about it. I think there's a lot of potential there. There are not many labs that do this and have this technology. At UW, there's significant expertise on this particular type of experimentation, and I think there's really a lot of excitement about this. So we're trying to push this to both thinking about how we could use the connectome to better develop interventions, but also how to connect that with function. And both of those are, I think, really cool questions to look at.
Thank you.
Of course.
Great, thanks. I will selfishly ask my question next then. So you talked about a lot, of course, explainability and causality being somehow tied together. Is the evaluation of a model through this lens of explainability another way to getting at the objective of trying to check whether the model is kind of truly capturing causal relationships? So is it just another way of evaluating causal versus spurious, or is it the intent and objective seem to be different, but is that kind of a similar tie?
Yeah, that's a fascinating question, and it's a topic of a whole different conversation that I'm happy to have, because I'm super excited about, I'm interested in that as well. But in very short, there's this whole notion of invariance that is related to causality. That basically the idea is that causal links are the only ones that are persistent in different environments. That if you look at prediction, the prediction might change in different environments. But if you have a causal link, if you have a causal relationship between A and B, regardless of the environment, that causal relationship does not change. And so there's a new class of ideas and methods that people are developing to actually infer causality based on this idea of invariance, to look at the performance, the predicted performance in multiple environments to see which ones are the ones that are consistently coming up, and using that in order to infer causal relationships. And I think that's actually, I mentioned briefly that there are ways to push beyond this issue of not having enough resolution over time, and that's actually one of them. So this whole notion of invariance is one approach that at least I'm hoping that we could use in order to use time course data to get at causal relationships.
Great, thank you. The next question is from Dr. Harefield. I think it's mostly asking, and William, you can come on if you're still here, unmute and ask the question if you want. But it sounds like it's basically the idea is there's the use of, for instance, Markov models to kind of reverse engineer and try to find explainability of existing, algorithms. He gives an example there. Has there been any way to kind of thinking about how to reverse engineer that kind of explainability by using something like a Markov model?
That's an interesting question. I don't know. I don't have a good answer for that. Whether we could use inversion basically in order to decide explanation. I mean, that inversion to me again gets to the causal question, and maybe if that's the goal of explanation, yes, that inversion by default and by the way that at least I understand it is going to get to the causal question. That gets to the question of whether A causes B, because that means that basically you could intervene on A and see a change at B. So I think if the explainability is more linked to the causal mechanism, then I think yes, that's one approach. That sort of inversion is one approach. I'm happy to talk more if this is not the right answer. Please let me know. I don't know if I'm understanding the question completely.
Thank you for that. My question is, in the simplest terms, if you're going to break it down, for instance, a human clinician could understand it, you would have 500 rules. If A, then B. If C decreases, then E increases. But I mean, that would be trivial and kind of ridiculous. But with as complex as the nets you're describing, surely there's some way when you make an observation to say, these nodes profoundly affected what we observed, in a way to identify what weights overwhelmed the decisions or the outputs that you observed.
Oh, thank you. Thanks for clarifying that. Yeah, absolutely. I mean, again, sort of what you're saying does seem the causal question, because then you're saying that sort of it impacts the output. And I think you're absolutely right. If we are able to get networks that are causal networks, so I talked about one example using spike train data that we might be able to get to causal networks, then yes, then we would be able to answer that question exactly the way you raised it, that if this neuron we're able to change it, we're going to see this output on the neurons that are impacted by it. So we have much more specific mechanisms that are causal. In the other cases, if we're not able to get it at the causal network, in the case of Granger causality that I mentioned at the beginning, it's not guaranteed to be causal. So it might still be predictive, but not causal. So we might not have that link. This is what I was saying in my response to Noah, that a little bit earlier, that if we are able to push it back out of this prediction and get to the point that we could say that this is the causal link, then again, we would be able to answer the question that you were asking in that scenario as well.
Great, thank you. Another question is a very important one, which is, when will the registration for the SISBID open? And maybe you could just give us a couple more comments about the requirements for the course and things like that.
Yeah, sure. So SISBID opens, registration opens early 2022. So the plan right now is to open registration in January. Usually that's when we open registration. We're right now updating our registration system. So I don't have an exact registration open date yet because the system is being updated. But my expectation is that we'll open registration in January. For folks that are at UW, there's reduced cost for registration for folks at UW, and there's early registration sort of costs. I mean, the modules are in general pretty reasonably priced. I mean, the prices are pretty low for basically academia setting. So these are shouldn't be a big factor, but even with that, because we're running it virtual, we've been able to run these at reduced prices, and for UW folks, that there's even a discount on top of that. So please take a look at the site. We will announce it once the registration is open, but my prediction is that it'll be by the end of January.
Great, thank you. I put the link there, at least the one that I think is the same kind of thing for from Google in the chat for people that want to click on there. It looks like there's going to be a lot of information coming out there. Are there any other questions? We're definitely nearing the time, but if there's time for one or two more questions. Let's give one moment to make sure. Great. Well, we had great attendance. I think that was a great talk. Thank you so much, Dr. Shojaie.
Thank you, Noah. Thanks for the invitation.
Great to have you. Yeah. And remember, everyone on the chat, in the call still, remember to sign up for our mailing list. We have great talks coming up, and we'll see you all there.
Thank you.
Thank you all.